comes from Cedar Falls, Iowa, although she was born in California. Correct. She arrived at Lawrence as a piano performance major in the conservatory, but pretty rapidly was drawn to the college and pursued her interest in science, her calling. She has many talents. I remember early on when I started advising you, you thought you might be a science writer at one point. But soon became possessed with the Ribonuclease Inhibitor, which in spite of its efforts to kill her, she has persisted and survived. And in fact is eagerly preparing for a career in some kind of biomedical research, possibly, probably cancer biology. So on and off for the past 3 years, Sarah's worked in my lab. She took a little hiatus and worked with Stefan doing some organic synthesis work, and then also took a trip to California for the summer and worked in cancer biology. And actually, no, the stem cell cardiomyocytes lab at UCSF for a summer. Has returned to finish her senior experience, also working more on the Ribonuclease Inhibitor. So, post-graduation, Sarah's going to be traveling to Boston to work at Mass Gen Hospital, part of Harvard, with Adam Feldman doing cancer biology. And with that, I give you Sarah.
Sarah also, I have to say, Sarah also helps elevate quality in the local brewing scene.
Oh, that's great. I forgot about that recent. You're right.
Yeah, that's right. Okay, thank you everybody for coming. Today I'm going to present the research I've been doing with Kim for the past 2.5 years, I guess now. And the title of my talk is Exploring the Ribonuclease Inhibitor's Implications in Cancer Through Its Purification. So our bodies have many mechanisms to defend itself against the development of cancers. And so in order for cancer cells to start tumors and grow metastases, they need to develop some traits that healthy cells don't have. And 6 of the most important traits that cancer cells have that healthy cells don't include the ability to sustain a proliferative or growth signal, so dividing without any sort of inhibition. They also need to resist programmed cell death, which all healthy cells experience when they become too old or aren't doing their job correctly anymore. Cancer cells also need to be able to evade growth suppressors that your body makes to defend itself against things like cancer from arising. They also need to be able to invade local tissues, but also faraway tissues to create secondary tumors and metastases. But most relevant to this talk is cancer cells' ability to induce angiogenesis. So angiogenesis is just the process of forming new blood vessels, and we generally think about this being turned on only during development and then during wound healing in a healthy adult. But for the most part, this is turned off. However, cancer cells depend on this process to form tumors because they need to recruit more blood than usual to form all of these new cells and provide all of these new cells with nutrients. So cancer cells need to make this transition from not forming any new blood vessels to forming new blood vessels in order to make these tumors, and this transition has been termed the angiogenic switch. This angiogenic switch has been the target for a number of pretty successful drugs that aim to prevent the formation of secondary tumors, but also to reduce the size of the primary tumor in the first place by cutting off its blood supply. So in order to target this with drugs, we need to make sure that cancer cells can't respond to the chemical messengers that they depend on to form these new blood vessels. And one of the most important chemical messengers that cancer cells make is VEGF. And VEGF is so overexpressed or made in excess during cancer that we can use VEGF to decide whether or not tissue is cancerous or not. So in order to target this angiogenic switch, we could prevent VEGF from having biological activity. If we do this, then angiogenesis can't happen and these tumors can't form. The problem with cancer is that everyone's is so different, so one drug target is never enough. And we need to learn a little bit more about angiogenesis if we really want to make sure that cancer cells can't turn this process on. Another protein that's important in angiogenesis is the protein angiogenin, which I will abbreviate to ANG for the remainder of this talk. ANG is critical in angiogenesis in that if we take ANG and we change or mutate it in such a way that it's no longer biologically active, then angiogenesis doesn't happen. We also know that angiogenin works upstream of VEGF, which just means that in the order of things, angiogenin gets turned on and then can go and cause VEGF to be turned on or activated as well. So maybe someday we can target ANG as a way to prevent tumors from turning this program on so that VEGF doesn't even get turned on in the first place. So angiogenin is just a ribonuclease, which is shown here in orange in complex with a protein that I'll talk about in a minute. So this angiogenin is a ribonuclease, which means its job in the body is to go around and cut up RNA. This is a really important activity to have on our fingertips and in the spaces between our cells where we need to protect our bodies from foreign invasion. But this would be a horribly detrimental thing to have happening inside cells where hundreds of thousands, if not millions, of RNAs are carrying out vital cellular processes such as protein synthesis. Without these vital cellular processes, cells wouldn't last for very long. So with such a potentially lethal role, it's really important that cells control angiogenin activity inside of the cells. And the way the body accomplishes this is through an aptly named protein, the ribonuclease inhibitor, which is shown in green in complex with angiogenin. So the ribonuclease inhibitor just makes sure that angiogenin can't act inside of our cells by binding to it and preventing it from cutting up RNA. So this is just an effort to protect all of our cellular RNA from angiogenin, as well as other ribonucleases too. So from a cell's point of view, it makes sense that this binding of RI and Ang would be extremely tight, because if Ang could get away from RI, it could go and wreak havoc and eventually lead quickly to cell death. So an overarching and main goal of this project is to better characterize this binding between RI and Ang. Maybe this binding is going wrong during some disease states, but before we can really even ask those questions, we need to learn a little bit more about RI, which has been left relatively poorly characterized. So RI is a cytosolic protein in all vertebrates, which just means any animal with a vertebrae has this protein in the liquid of its cells. This is a good place for RI to hang out, since it's the most likely place that angiogenin could come in and eat up all of its functional RNAs. RI structure is comprised of 15 leucine-rich repeats, which are made up of a beta strand, a turn, and an alpha helix. And there are repeating patterns of these between A and B type, which just differ by the number of amino acids in each motif. So this structure allows RI to be highly symmetrical, but also very flexible. And if we flip RI down 90 degrees and we see its core is hydrophobic and has many beta strands that align in a parallel fashion, that allows RI to afford a lot of surface area for the binding between it and other ribonucleases. So because of this leucine-rich repeat structure, it allows RI to have extremely high affinity for the RNase superfamily, not just angiogenin alone, which is actually quite surprising. To give you an idea of how tight this binding is, the equilibrium constant for this complex is 7.1 times 10 to the negative 16th molar, which essentially implies once these proteins come together, they're not going to come apart. Many people think that the biotin and avidin complex is the tightest complex in nature, but actually the equilibrium constant for that is 6 times 10 to the negative 16th molar, which implies that the binding of RI and Ang is actually a little bit tighter than that. So this is truly one of the tightest bindings in nature. So in order to study this more and maybe how it could possibly get dysregulated during disease states, we need to get a quantifiable and studyable amount of RI in the first place. So one of the main goals of this project that I've spent a lot of time on has been optimizing a protocol for the expression and purification of RI. This has turned out to be a lot easier said than done due to a little detail about RI's structure that makes it incredibly difficult to work with. And that is that RI is very sensitive to oxidation, and this is due to the 32 cysteine residues that are found in human RI. So these residues are shown in a color code scheme in these spheres in the tertiary structure of the protein, and they're just color coded by where they fall within its structure. So the green are all located in the alpha helix, whereas the orange are more in the turn. This 32 cysteines are actually a really high number. On average, most cytosolic proteins have a cysteine content of 1.6%, but these actually make up 7% of RI's total structure. So these cysteines are just amino acids, and two residues are shown in this diagram here. In the reduced conformation, cysteines have a free thiol group, but when they come even remotely close to each other, they quickly form a disulfide bond, which leads to an increase in the oxidation state of the molecule as a whole. So this reaction happens really, really easily, and even the introduction of a lot of bubbles during certain steps of the process will lead to this reaction. Unfortunately for us, all of these 32 residues need to be in the reduced conformation for RI to have any sort of inhibition activity. So for RI to contact any of its RNase ligands, it needs to be completely reduced, which implies that these are probably contributing to the structure of RI. In order for RI to be propped up in this horseshoe, it needs to have all of them reduced, and once they start to oxidize, it collapses and can't have any of its surface area contacting these ribonucleases. So RI would be so much easier to study if it wasn't so sensitive to oxidation. And so one of the other goals of this project has been to design and purify variations on this protein that contain changed cysteine residues that we change to other amino acids that don't have such high probability of oxidizing. So if we go into the DNA sequence of RI, and we can change it such that maybe we change all of the orange cysteines to other amino acids, and this would leave us with a variation on the protein that hopefully wouldn't be so susceptible to this oxidation and inactivation. We might change all of the blue cysteines to other amino acids, and in total, we generated 7 genes that have variations on RI. In one of our proteins, we actually took out all of the cysteines. So this cysteine-free variant probably will have a different amount of activity that it can bind Ang with, but hopefully the structure won't be so compromised, because we hope to find some order of taking out these cysteines that lets RI still bind and inactivate Ang and still have its horseshoe-shaped structure. So the point of these mutagenesis studies are to eventually understand why those 32 cysteine residues are there. I already said that these cysteines are definitely contributing to the structure of RI, but because all vertebrates show this high sensitivity to oxidation and they continue to evolve with a lot of cysteine residues, it seems likely that these cysteines are playing some other role in RI. Maybe RI is serving some other purpose in the cell by being so sensitive to oxidation. A perfectly reasonable question to ask is, how sensitive are these to oxidation inside a cell? Is this sensitivity just something that scientists have to worry about in a lab, or do cells have to worry about oxidation? And the short answer to that is no, it's not just something we have to worry about in the lab. Cells definitely have to worry about oxidative stress. In fact, every time cells undergo any sort of activity, whether it's metabolism or respiration, every time you exercise, your cells produce tons of these oxygen species that are potentially harmful to cells. They're harmful in that they can go around and damage DNA and proteins. And while healthy cells can protect themselves against these species, just as a car rusts after its steel has been exposed to the elements for long enough, enough oxidative damage leads to aging, and a lot of aging theories are based on these oxidative species. So these species don't just contribute to aging, they contribute to a lot of disease states, and cancer is only one of them. But cancer is tricky because while it might arise from this stress, it also contributes to this stress in a cycle that cells can't really escape. So it would make sense for cells to have some sort of sensor inside of them that would tell them, hey, there's too much oxidative damage. We better undergo programmed cell death rather than hang around this body and maybe contribute to something like cancer. And so RI is in a pretty good position to play this role since it's so extremely sensitive to oxidation and it plays such a vital role. We can imagine as more RI is inactivated from oxidative stress increasing from a number of mechanisms, more free RNases can go around and chew up your functional RNA. Once your functional RNA starts to get chewed up, cells would quickly die and life wouldn't continue for very long, which in this case, again, would be preferable than hanging around and contributing to some disease state. So to study all of these questions we have about RI, we first need to get enough of it to study. And so the first step in getting enough of protein is convincing E. coli to make enough of it for us. So this could be our wild type or normal RI, or it could be any of the variation proteins that we've made. We then need to make sure that we have only RI and that it's not mixed in with other proteins that E. coli are going to make. We also want to take RI out from all of the E. coli that made it. So once we've separated the RI, we can move on to some analytical steps, such as assessing how well this protein that we made can bind angiogenin or other ribonucleases. We want to make sure that when we change those cysteines, we didn't mess up the structure or how well that horseshoe could form. And finally, and eventually, we want to study this interaction between RI and Ang, and maybe if it changed when we changed those cysteine residues, we want to know why. So to first make sure that E. coli can make this RI, we have to program these cells like a computer. So computers read code, and E. coli read a code in the form of a plasmid, which is just a circular piece of DNA that has certain traits that we can control and tell E. coli how quickly to make this protein for us. So the plasmid we buy from a company, and it's actually really important which plasmid we decide to buy. Eventually we can put the isolated RI gene or RI variant gene into this plasmid to create a construct which will be introduced to these E. coli cells. E. coli can then read the construct and make this red gene into functional protein for us. As I mentioned, that this plasmid that we choose is really important, and so this expression vector we've recently switched to, and it has a number of characterizations that I want to point out. The first is that we're inserting RI downstream or attaching it to a protein called SUMO, which is a small protein that's going to let RI remain soluble during its expression. So something we worry about during protein expression is whether or not the protein that we make will be soluble, or if it will just end up unfolded in weird clumps that we can't get RI back out of, called inclusion bodies. So if RI goes into inclusion bodies, that's sort of the end of the road for us. Another thing that I want to point out is that this vector has a his tag shown in blue, and this his tag is going to let us pull RI out from all of the other junk that E. coli is going to make. Finally, this gene segment is all under the control of a rhamnose promoter, which is actually in contrast to what expression vectors usually use, which is a T7 promoter. So T7 promoters are derived from a virus and are notoriously leaky in that they will express your protein even when you don't want it to be made. These promoters are also less like a faucet and more like an on-off button in that you can't really tell it how quickly to make your protein. It sort of just spits it out at whatever speed it wants. So the rhamnose promoter has allowed us to have a lot more control over how quickly our protein is being produced, and therefore how more likely it is to remain soluble. So after we've convinced RI to be made from these E. coli cells, we need to separate it from all the other proteins that these cells made. And so this has to do with that his tag that I mentioned just a second ago. So the his tag is acting like a handle that's being attached to RI for some molecular hand to grab onto. This molecular hand is represented in lime green here, and the hand is being attached to a column, which means it's not going to go anywhere, it's not going to be lost in the liquid anywhere. So once we wash RI over this hand, RI will be grabbed onto, but nothing else will be. Eventually, we can make that hand let go of RI by giving it something it wants to hold on to even more. So this technique is referred to as immobilized metal affinity chromatography, where chromatography just means separation. And metal in this case is nickel, and this nickel is acting as our hand. So the hand is being tethered to this column, and the hand can grab onto RI's affinity tag, which is shown here as almost a tail hanging off of RI. This hand can only grab onto the tail, so nothing else gets purified or caught by this hand, and eventually we can tell it to let go by giving it something with even higher affinity, something it can hold onto even better. We can remove this affinity tag from RI if we really want to in a little while, but this is an image from a protein gel, and while this is messy, it does actually show something of interest. So all of the blue dye that you see are indicating that there's protein there, and these gels are read through the biggest proteins being stuck on the top and smaller proteins coming down towards the bottom. And so in order to read this, we need to have some sort of marker to tell us how big certain proteins are within this gel. So in the first line, I have the molecular marker, molecular weight standard, and I've marked where 50 kilobases is. So everything in the same column as 50 kilobases, which is right about here, will all weigh about 50 kilobases. And I guess the next lane is the whole lysate, which is everything that we get once we explode an E. coli cell and all the protein that we get. After that is the clarified lysate, all of the soluble fraction where we hope RI is, and the insoluble stuff where all of the inclusion bodies and gross stuff out of a cell that we don't want coming out will be. The two wash lanes, or W1 and W2, show when we've washed all of the junk off of the hand, this is what came out. And eventually the elution is when we've told the hand to let go of RI. I want to point out that there's two bands here with significance, and I know it's hard to see, so you might just have to take my word on this, but there's a band in the whole lysate right above 50 where we expect to see RI, and this band doesn't show up again in the washes until the elution one, which means we truly captured RI with that hand and then got that hand to let go of it. While this is only preliminary data, it's really solid confirmation that our expression and purification system is working. So we can then use this eluted protein, or stuff that the hand let go of, to do our analyses, which include first assessing how well this protein can bind to angiogenin or RNase A. And so eventually with these we're going to be able to calculate a value that we use in biochemistry called the inhibition constant that's going to give us an idea of how much RI it takes to inhibit RNase A's ability to cleave RNA by 50%. We can compare this Ki between our wild-type protein and the variation proteins that we made, and then eventually we want to make sure that the variations that we made truly are less sensitive to oxidation. We're going to do that by inducing oxidation by introducing hydrogen peroxide to the system. This hydrogen peroxide will oxidize wild-type protein for sure and leave it inactive, but hopefully one of our variants will be less sensitive to this oxidation and retain ability to inhibit RNase A. So these activity assays depend on a technique called FRET, which stands for Forster-Resonance Energy Transfer. And the way this works is by tying together two fluorescent molecules with a piece of RNA. This RNA is susceptible to cleavage by RNase A, which can go around and cut any RNA that it sees. So this works if we shine a light at FAM, it will be able to transfer light energy over to TAMRA. And once TAMRA gets that light energy transferred, it will emit orange light. This is in the scenario that this RNA piece is intact and hasn't been cut yet. But if it does get cut, when we shine this light at FAM, instead of transferring light energy over to TAMRA, it will instead just emit purple light because it won't be able to translate this light over once that RNA has been cut. So this only matters to us. I guess the only significance is if we detect for purple light in the bottom scenario, we're going to get an increase in signal. But if we detect for purple light in the top scenario, we're actually not going to get any increase in signal because there's no purple light being produced. So if we add RNase A to this system and we detect for purple light, we're going to see an increase in signal. But when I add RI to this system, we hope that it will complex with RNase A and that purple signal won't be able to be generated since RNase A won't be cutting the RNA that would lead to that. And so that's exactly what I've observed in this preliminary activity study. So I want to point out that the y-axis is just detection of counts or how much signal is being produced, purple light, and the x-axis is just over time. So between points A and B, we see an increase in signal, which indicates that RNase A is going around and cutting that RNA between those fluorescent molecules. Between points B and C, we don't see any increase in signal. In fact, there's no signal being produced, which indicates that RI is active and that it's complexing with RNase A and not letting it cut that RNA. At point C, I've added an overwhelming amount of RNase A so that RI can't inhibit it, and we can make sure that this inhibition between B and C is really due to RI and not just due to RNase A running out of RNA to cut. While this is only wild type, again, this is really promising because it means that the protein that we've made from our new system really is active and soluble and not just the same size as something we're looking for. So once we've assessed the activity, we can move on to more characterization of what that protein structure looks like. And the rest of these data I'm going to show come from an older vector that we're no longer using. So what is structural integrity anyway? Well, when we talk about structural integrity, we're asking how well our protein was able to fold. So all proteins need to take on a certain conformation that allows them to have whatever action they do have. And there's a number of factors that can push a well-folded protein over to denature or unfold and just become a tangled mess of inactive amino acids. So one of these factors that will push that over is heat, and as we heat up proteins, they denature and they unfold. So all proteins have a certain TM, or thermal melting point, at which point they denature, and the determination of this can allow us to understand how well our proteins have folded. Maybe they're only halfway in between folded and denatured, which would be bad. But the way this is determined is through a technique called differential scanning fluorimetry. The way that this works is using a hydrophobic dye that will bind to hydrophobic residues and fluoresce. And so hydrophobic residues are really like the guts of a protein, in that when they're folded up, hydrophobic residues are held inside, but when they denature, all of their guts spill out, at which point that dye could bind onto all of the hydrophobic residues that it exposes and fluoresce. So at whatever temperature it is that these proteins denature, we should see a peak in fluorescence that indicates they've denatured. Another way we can characterize this is through circular dichroism spectroscopy, which I'll detail in a minute. And so again, these are just going to allow us to answer how changing those cysteine residues affected the structure and ability for RI to fold. So these are the melt curves for our wild type and variant proteins. On the y-axis is how much fluorescence is being detected, and the x-axis is temperature. Just like I said, we see a peak in fluorescence that corresponds to the temperature at which point these proteins denatured. I want to point out that there's a couple curves here that are actually shifted to the left in comparison to all of our other proteins, and this corresponds to a lower TM actually, and it turned out that these proteins with a lower TM were the RI standard that we had bought from a company. So this company purifies RI from pigs and then sells it to labs for a really high cost. And so we know that we're purifying human RI, which has some structural differences than pigs. So maybe this TM was due to that. And also, differential scanning fluorimetry had never been applied to RI, so we weren't really sure what to look for anyway. So for these reasons, this discrepancy wasn't too concerning to us yet, so we continued down the path of structural characterization. This led us to circular dichroism spectroscopy, which relies on the basic principle that different structures rotate and absorb light differently, and that proteins are made of two essentially different structures, alpha helices and beta sheets. And so if we shine circularly polarized light at an alpha helix, that's going to rotate and absorb light in such a way that this data will give us something that looks like the yellow curve. So the y-axis is just a measurement of how much this light has been rotated from circular more to an ellipse, and this is a function of wavelength, so that's being reported on the x-axis. If we shine this light at a beta sheet, it's going to rotate light in such a way that gives us data like the blue curve. The red curve is just a random coil or unstructured piece of amino acids. So this technique allows us to determine the amount of our protein that is either alpha helix or beta sheet, and so just like everyone has a fingerprint, every protein would have their own CD spectrum. Going into CD, you might have a pretty good idea of what the spectra should look like if you know what your protein structure looks like. So we know RI has a lot of alpha helices, but we also know it has a lot of beta sheet quality to it. So we expect to see some CD spectrum that looks like a mix almost between the yellow and the blue curve. When we got the data back, this pretty much implies that whatever protein this is mostly alpha helix, and there's really not very much beta sheet characteristic to it at all, which was a red flag because RI really should have quite a bit beta sheet to it. So if this protein that is being plotted here wasn't RI, what protein did we purify then? Well, if you remember, I've mentioned a couple times that we've recently switched to the vector that we're using now, where we've attached RI to SUMO and also to a his tag. But before we were doing that, we had been attaching RI to something called maltose binding protein, which served both to help RI be soluble, but also to purify RI. Instead of a his tag, we were using maltose binding protein. So to truly purify this, we need to make some cut between RI and maltose binding protein at this blue junction, and unfortunately it seems like when we made that cut, we ended up purifying the wrong end of the fusion protein, because the published data for maltose binding protein looks exactly like what we got. So we purified a protein that no one cared about at all. And this was a really big setback and highlighted the in research for everybody involved. And so we haven't been able to study RI with Angiogenic. We got all the way set back to convincing E. coli to make RI, but we can still make some conclusions from this. And these are that genetic variants of RI can be purified and they are active. I know now that RI can be made from a rhamnose promoter in fusion with SUMO and IMAC purification. We also now know that the vector that we had been using not only was giving us poor purification products, but also was causing us a lot of problems throughout the whole cloning sequence. So this vector was probably faulty and maybe contaminated with something we don't really know. But these future directions, of course, now include cloning our variant genes into our new vector, comparing these activities across variation and wild-type RI. We also want to get accurate structural data for now even wild-type RI, and to eventually elucidate RI's role in the angiogenic switch. So I want to thank my advisor Kim, of course, for all of her help, as well as Alec Uberson for putting in a lot of work, and Wynn Joanne in the stockroom, of course, and Dr. Stork for his technical help during the FRET assays. And everyone for coming. Okay. Does anyone have any questions?
Am I moderating questions or are you moderating questions? Or does it matter?
She moderates the question.
You moderate questions,
Right?
awesome.
She just points.
Yes, Sarah.
So when you purified the RI and you had that really light band, how are you are you trying to come up with ways to enhance the amount of RI?
Yeah, and so that optimization of the protocol includes making sure that that band matches sort of the band that we see in the whole lysate. And so clearly hasn't been optimized yet, but that's certainly confirmation that maybe someday it will be optimized. Yeah. Oh, Dr. Stork.
On the bell curve, you had on the y-axis normalized reporter.
Yeah.
I wonder what was it normalized to?
That's a really good question. I'm not sure. I think that the qPCR might just do that on default, or maybe you have to normalize it to whatever buffer it's in.
Okay, because I was looking at the traces and they were normalized.
Right, yeah, I'm not sure. That data is from a couple years ago now, I guess. So I'm not sure what it was normalized to.
Can you go back to that figure?
Yeah, and there wasn't data in there actually that had them all normalized. So if someone did that, then that was not in the qPCR program.
Yeah, I think that's just die-in buffer, as I recall. There's no normalization reporter that you use in qPCR for this kind of assay.
Oh, yeah.
So the data you got from the FRET assay, was that using recent RI
Yeah,
that you
so
know is actually RI, not maltose binding protein?
Exactly,
Okay.
and so that is from the new vector where we've thrown all of this stuff away because it's just not going to work. And so that FRET data is from the new stuff used with SUMO and the his tag. And that little band that you saw in the gel, so that band that you see actually is active even though it's very minimal in amount.
And this new reporter is supposed to increase expression as well? Is that right?
The new vector,
Is it supposed to increase expression of RI?
I mean, hopefully that we can get more RI out of it than we had been. Hopefully it will increase the amount of soluble RI that we're getting because it's not just coming out like at light speed. Now we can have it sort of drip out. Yeah.
What happens if you put some of your RI variants into mice, for example? Do they die, or?
Well, hopefully not.
Well, has anybody tried that? Like,
No.
change the system?
In mice?
Yeah, or
Well-
in anything that has RI, what happens if you get rid of a couple of the cysteines?
Oh, I don't know if those studies have been done at all.
No one's reported of RI mouse knockout yet.
I imagine knockout would be dead, but I was kind of interested in like the cysteine, get rid of a few cysteines, because you were mentioning the redox potential is really important, and so if you get rid of them, you change redox potential, and then, I don't know, mouse dies of cancer, mouse ages prematurely, mouse is perfectly fine with wavy hair.
I think that's kind of a long way down the road, maybe at this point in terms of RI's characterization.
Not in our lab for sure.
Yeah, it's really not going to be me.
Questions?
Could you go briefly over how the thread assay works one more time? How the energy is transferred and so on?
How the energy is
And the
transferred?
light.
That's a good question. I definitely don't know the physics of
This
this.
can be the broad, because I'm a layperson here.
Oh, okay. Well, when they're connected with that piece of RNA, these fluorescent molecules, they can transfer this light energy through that RNA, through, I guess, electron transfer, or if that's not true.
It's just close proximity.
Through space.
Through
Yeah,
space?
through space.
Okay. And so I guess then when that's cut, then they float far away and they're not close anymore. So it's easier than that.
So it's kind of as a general thing to see how close two things are.
Okay, let's thank our speaker one more time.